Steps in formation of recombinant DNA by action of restriction endonuclease enzyme. Restriction enzyme occurs naturally in certain bacteria and act as defense mechanism against virus. Each restriction enzyme cuts at a specific location, recognition site, where a particular sequence of nucleotides is found.
The restriction enzyme EcoR1 has the following recognition site:
GAATTC, CTTAAG. This means that
EcoR1
will only cut between G and A in this sequence. Step 1. A section of DNA with a recognition site for
EcoR1
is provided. Step 2.
EcoR1
Cuts the DNA strand at the recognition site between G and A. Step 3. The DNA strand is fragmented. This produces DNA fragments with exposed nucleotides or sticky ends. Other restriction enzymes, for example HIND2, make a straight cut and produce DNA fragments with no exposed nucleotides. These fragments are called blunt ends. Sticky ends and blunt ends can be joined together by another DNA enzyme, ligase, in a process called ligation. DNA ligase is involved in the repair and replication of DNA. Ligases function to rejoin the DNA fragments in a process called ligation. The DNA fragments join together using the base pair ruling. The exposed nucleotides of a fragment of DNA
are attracted by base pairing to another DNA fragment.
The fragments are linked by the enzyme DNA ligase, which joins the fragments at their sugar phosphate backbones. If the DNA fragments are from different sources, recombinant DNA is produced.
For more such videos on all subjects, practice questions and a lot more, download iPrep and learn unlimited.